Okay, so welcome back to this next video in which we are discussing drug addiction. Okay, so we're currently working through many different examples of addictive substances and seeing how all of them work to increase dopaminergic transmission within the nucleus accumbens and the medial prefrontal cortex. Okay, and how, well, what we know is that when dopamine goes up in the nucleus accumbens and the medial prefrontal cortex, what that is doing is it's a teaching signal. It says, right, we now need to remodel the brain, and we're going to remodel the brain so that you start craving and wanting these addictive substances, the substances which produce the rise in dopamine. And secondly, so that we can forge memories, specifically forming associations between cue-related stimuli, okay, and then the relation between that and then initiating the drug-seeking behavior. Okay, right, so what we now want to move on to is the benzodiazepine drugs, okay, which are drugs that are used as anxiolytics, which means that they are used to treat anxiety, okay, they make anxiety better, okay, and they're also used as hypnotics, which means that they help you get to sleep, okay, they make you sleepy. Right, but they can also produce addiction, and this is one of the things that curtails their use as anxiolytics and hypnotics. Okay, right. So firstly, let me give you some examples of benzodiazepines. In fact, actually, before I give you examples of benzodiazepines, let me tell you what chemical structure all benzodiazepines have within them. Okay, so they all have what's known as the benzodiazepine structure. Okay, so I'd like to firstly show you the structure. So, the benzodiazepine structure then. So firstly, it consists of what's known as a 1,4-diazepine ring. So I'm going to show you the 1,4-diazepine ring first, and then we're going to add a benzene ring onto the 1,4-diazepine ring, and that will create us a benzodiazepine structure. Okay, so a 1,4-diazepine ring is a 7-membered ring structure with a nitrogen right at the center and a second nitrogen at the 4th position, okay, so this is the nitrogen at position 1, okay, and all the other 5 members of the 7-membered ring are going to be carbon atoms. Okay, so here's a carbon atom here, okay, there's another carbon atom there, okay, so that's 1, 2, 3, 4, 5, okay, and then we need 6 down here, 7 down here, and then we're going to just join those two together, and there's our 7-membered ring. 1, 2, 3, 4, 5, 6, 7. Okay, now, whoops, this should have been the nitrogen rather than the carbon down here. I apologise for that. Okay, so this is position 1 of the ring, 2, 3, and then this is 4, which is why it's called a 1,4-diazepine ring. So this tells you where the nitrogens are in this diazepine ring. Now the other thing that I haven't put on yet is the fact that to be a diazepine ring, it needs to have 3 double bonds. 1 here, and 1 here. Okay, right. Now you can have other sorts of diazepine ring, okay? Then this nitrogen is always in the same place, okay? It's always number 1 up there. But you can have the second nitrogen here rather than down here, and you'd have a carbon down here. That would be a 1,2-diazepine ring, okay? Or you could have it here, which would be a 1,3-diazepine ring. And you might say, well, what about over here and over here? Well, if it was in one of these 3 positions, that would be equivalent to it being in the symmetrical position on this side. So there are only 3 different types of diazepine ring. 1,2-diazepine rings, 1,3-diazepine rings, and the 1,4-diazepine ring that's going to be present within the benzodiazepine structure, shown here. Okay, now, to turn it into a benzodiazepine structure, what we need to do is stick a benzene ring off the side here. Okay? So here is our benzene ring off the side of the 1,4-diazepine ring, and that entire structure now, that's known as the benzodiazepine structure. Okay, so all benzodiazepine drug molecules are going to have one of these benzodiazepine structures within them. Okay, right, so let me give you some examples of benzodiazepine drugs then. So the first one that I cannot possibly not say is diazepam. Okay, so this is a very, very famous drug. It's more famously known by its first ever trade name, which was Valium. Okay, so yeah, very, very famous drug. Okay, other drugs, the first ever benzodiazepine was chlordiazepoxide. Okay, so I'll also mention that one. Diazepoxide. Okay, and then two more. There's also lorazepam. Okay, and also clonazepam. Okay, so you'll notice that a lot of them have the ending zepan. Okay, and that's a way that you can distinguish them. Okay, right. So there are 4 examples of benzodiazepine drugs. Okay, what we now want to see is how are these drugs going to result in an increase in dopaminergic transmission within the nucleus accumbens and the medial prefrontal cortex. Okay, right. Now, in order to see that, we need to know something more about the target for these drugs. Now, the target for these drugs is the receptor for GABA. Okay, so we've mentioned this many, many times. Okay, we've talked a lot about GABAergic neurotransmission, and GABA, this inhibitory neurotransmitter, acting on ligand-gated ion channels, which then open and allow chloride anions into the cell. We now need to discuss the structure of those receptors for GABA in a little bit more detail. Now, before we discuss the GABA receptors, I would just like to, for completion, talk a little bit more about what GABA is. So GABA is the main inhibitory neurotransmitter that works mainly on ionotropic receptors. There are some G-protein coupled receptors for GABA, but they are much, much rarer compared to the ionotropic GABA receptors, or the ligand-gated ion channel GABA receptors. Okay, now, what does it stand for? Well, it stands for gamma amino. Okay, and if I wrote out gamma properly, it would begin with a G there, so gamma amino. So the first G in the name is for gamma here. Okay, so that's gamma. Okay, then the A, the second letter, is for amino. Okay, and then the final BA is for butyric and then acid. Okay, so the B is for butyric, and then the A is for acid. Okay, now, this neurotransmitter actually has an extremely simple structure. Okay, so as soon as you know what butyric acid is, you can pretty much work out this molecule. Okay, so butyric acid is just the old name for a molecule that would nowadays be called butanoic acid. Okay, now butanoic acid is just a 4-carbon carboxylic acid, so let's draw this out here. So here is our carboxylic acid group, okay, and then we need 3 more carbons to take us up to a total number of 4, okay, and then off these carbons we're going to have hydrogen atoms, one here, another one here, another one here, another one here, another one here. Now, if we were just drawing pure butanoic acid, I would put another hydrogen there, okay? But we want gamma amino butyric acid, but remember, butyric acid is just another name for butanoic acid. Okay, so we want an amino group on the gamma carbon. Now, this is an old way of naming the carbons in a carboxylic acid. Okay, so the old way of naming the carbons in a carboxylic acid was to name the first carbon that comes off the carboxylic acid carbon, to call that the alpha carbon. Okay, so let me get some pens. So this one here would be the alpha carbon, and then you'd continue on basically down the Greek alphabet. Okay, so the second one here would be the beta carbon. Okay, this one here. And then this third one here then would be the gamma carbon. Okay, so what we want is an amino group off the gamma carbon to create gamma amino butyric acid. So here's the amino group coming off this gamma carbon like so. Okay, and this molecule now, this is gamma amino butyric acid. Okay, the main inhibitory neurotransmitter of the brain. Okay, now, we want to discuss then the GABA A receptors. Okay, so remember, when we had an inhibitory neuron like so, releasing GABA onto our dendritic spine, the GABA was going to act on receptors that were on the dendritic spine, okay, and these receptors were going to be ligand-gated ion channels, which were going to allow chloride anions into the cytoplasm of the cell. Okay, now these receptors are known as the GABA A receptors. Okay, there are also the GABA B receptors. The GABA B receptors are the G protein coupled receptors for GABA. Okay, so we now want to study the GABA A receptors in more detail. Why? Because benzodiazepines are going to work on these GABA A receptors. Okay, so let me firstly draw a picture then of a GABA A receptor. So let's let these two lines here represent the inner and outer, the outer and inner leaflet of the phospholipid bilayer of the postsynaptic membrane. Okay, so this side, this is the synaptic cleft. Okay, so this is the side where the GABA is being released onto us. Okay, and I've missed out the L there. Okay, and this is the cytoplasmic side on this side. Okay, and this is then the cell membrane sitting in between. Now, our GABA A receptor is going to be sitting in the cell membrane, like so. Now, whenever you're drawing a ligand-gated ion channel, it's always a good idea to start by drawing a cylinder. Okay, so here is our ligand-gated ion channel here, sitting in the cell membrane, like so. Okay, now, long ago when we were talking about dopamine receptors, I said a one-liner, which was that ligand-gated ion channels, okay, were not made up from a single protein. Instead, they were made up from multiple proteins stuck together. Okay, now that's going to become important now that we're studying the GABA A receptor here. Okay, GABA A receptors are all going to be made up of 5 separate proteins stuck together. Okay, like so. So they're going to be pentamers, okay, with 5 separate polypeptides all bound together to make up the full structure here. Okay, so this is our pentamer here. Okay, right. So what I now want to do is pull out one of these GABA A receptor subunits, as it's called, okay, one of the individual polypeptides that makes up a fifth of the GABA A receptor, and have a look at its membrane spanning topology. Okay, have a look at its basic structural features. So this is what we would call a GABA A receptor. So we'll put GABA AR for GABA A receptor, and then it will be called a GABA A receptor subunit. Okay, it makes up a fifth of the entire GABA A receptor, which is the full thing. Okay, right. So we'll need another piece of paper. Okay, so we're going to pull out that GABA A receptor subunit and have a look at its basic structural features. Okay, so it's an integral membrane protein and is therefore going to be spanning the membrane multiple times, or at least, well, to be an integral membrane protein, you need to span the membrane once. Okay, these GABA A receptor subunits are actually going to be spanning the membrane multiple times. Okay, so let's say that this is the amino terminus of this GABA A receptor subunit polypeptide, and then this line represents the polymer of amino acids. Okay, now extracellularly, okay, and I should have probably just said that this is the extracellular fluid side, or the synaptic cleft side of the membrane once again, and this is the cytoplasm side. Okay, now the amino terminus of the polypeptide starts off extracellularly. Okay, now one of the key structural features that you have long before you've ever begun the membrane spanning alpha helices is a structure known as a cis loop. Okay, now this is basically a loop in the polypeptide structure that is held together by a disulfide bond between 2 cysteine residues, okay, on the 2 parallel sides of this loop. Okay, so let me now talk you through this structure in a little bit more detail. Okay, so I'm going to draw it out bigger down here. Okay, so this line represents the polymer of amino acids again, so this represents amino acid after amino acid after amino acid after amino acid. And now I'm going to draw one amino acid much bigger than the others. I'm going to actually draw out its structure in full. And this is going to be the cysteine amino acid that's going to be involved in this disulfide bridge that's going to hold the cis loop together. Okay, so here's the amino group then of the cysteine amino acid. Here's the alpha carbon with a hydrogen coming off, and here is the carboxylic acid group here, which will be bound to the amino acid after it. Okay, and then the R group of a cysteine residue consists of a methylene group, like so, then with a thiol group coming off it. Now, a thiol group consists of a sulfur atom bound to a hydrogen atom like this. So this is a thiol group. Now, if you're not familiar with the chemical properties of a thiol group, then the thing that I can probably say to set your mind at ease is that it has very similar chemical properties to the alcohol group. Okay, and the reason for that is that sulfur here is in the same group of the periodic table as oxygen, okay, which means that sulfur atoms are going to have similar chemical properties to oxygen atoms. Now, if we replace that sulfur atom with an oxygen atom, then it would be an alcohol group, okay? So, since sulfur has similar chemical properties to oxygen, we might expect the thiol group to have similar chemical properties to the oxygen group, and indeed they do to an extent. Okay, right. Now, I have not shown the hydrogen coming off this sulfur atom. In a pure cysteine residue, there would be a hydrogen coming off that sulfur atom. Okay? However, I'm being fortuitous because this cysteine residue is going to be involved in a bond to another cysteine residue. Okay, and this bond is going to involve the thiol group. Okay, and we're going to lose the hydrogen off the sulfur atom of the thiol group. Okay, so that's why I'm not showing the hydrogen coming off that sulfur atom. Okay, now we'll go back to representing the polypeptide by a line here. Okay, now we're going to loop around, and there's going to actually be 13 amino acids in this loop here before we then get back to the next cysteine residue, which will be over here. So here's the amino group, here's the alpha carbon, so this is now the cysteine residue on this parallel strand. And as I say, there are 13 amino acids in between this one and this one. So should we actually put that in? Here's 1, here's 2, here's 3, here's 4, here's 5, here's 6, here's 7, here's 8, here's 9, here's 10. We're going to have to be careful. 11, 12, 13. Okay, so those are the 13 amino acids in between the cysteine on this side of the loop and the cysteine on this side. Okay, and then on the other side, here is our next cysteine. So here is the amino group, here's the alpha carbon with the hydrogen coming off it, here's the carboxylic acid group, and then you'll go into the rest of the polypeptide onwards here. Okay, and then the R group of this cysteine residue consists of a methylene group, like so, then with the thiol group, and now what's going to happen is both of these thiol groups are going to lose their hydrogens, and the two sulfurs are going to bind to each other rather than to hydrogens. Okay, and this bond here, this is what is holding the entire thing together. This is what's known as a disulfide bond, also sometimes called a disulfide bridge. Okay, and it is a very strong covalent bond that is going to hold this loop in the polypeptide structure together. Now, it is formed between two cysteine residues. Now, the 3-letter code for a cysteine is cis. Okay, hence the origin of this loop structure being called a cis loop. Okay, right. So, all of the GABA receptor subunits are going to have a cis loop in their extracellular portion. Okay, and I say all of the GABA receptor subunits, there are many different types, okay, and that's going to make GABA receptors, unfortunately, rather more complicated, because it is not the case that there is just one GABA receptor. There are a huge number of different types of GABA receptors. Okay, but we'll come on to that when we come on to it. Okay, right, now the polypeptide is going to span the membrane for the first time here, then it's going to turn around, span the membrane for a second time here, turn around, span the membrane for a third time, then you're going to have a large intracellular loop between the third membrane spanning alpha helix and the fourth membrane spanning alpha helix, and you're then going to finish with the carboxylic acid terminus of this polypeptide extracellularly. Okay, so let's highlight up some special features on this structure. So firstly, we have the 4 membrane spanning alpha helices here in vivid purple. Okay, and these are labelled up. M1 is the first one, then the second one will be called M2. Okay, so this will be called M2. Third one will be called M3. Okay, and then the fourth one will be called M4. Okay, right. So those are those membrane spanning alpha helices. Okay, we then have this large structure here between M3 and M4. Now there are various names for this. You can either call it the M3-M4 loop, which makes sense, okay, or you can call it the ICL. Okay, and the ICL is short for the intracellular loop. Okay, so I for intra, C for cellular, and then L for loop. Okay, now, this intracellular loop is the site of many of the post-translational modifications that occur to the GABA receptor. So, for instance, things like phosphorylation can occur on this intracellular loop, which can moderate the function of the GABA receptors. Okay, now, the other feature we've already discussed in detail, which is the cis loop out here, but I would like to say just one more thing about the cis loop, the cis loop, okay? GABA receptors is a massive great family of loads of different types of receptors, okay? And we're going to come on to all of the different types of subunits for GABA receptors in a moment. However, the great family of GABA receptors is in a larger family of receptors, okay? It is within the superfamily of cis loop ligand-gated ion channels. So there is a bigger family that contains, as a subfamily, the GABA receptors, and this is known as the cis loop ligand-gated ion channels. Okay, the cis loop ligand-gated ion channel family. Okay, now, this contains loads of different families of receptors, like the GABA receptors, which also have the same basic structure for their subunits. Okay, in fact, cis loop ligand-gated ion channels are all pentamers, okay, so they're all made up of 5 separate proteins stuck together, and the subunits of those pentamers all have this basic structure where you have the 4 membrane-spanning alpha helices with the amino and carboxy terminus on the extracellular side. You have a cis loop extracellularly prior to M1, and this intracellular loop between M3 and M4. Okay, so the GABA receptors, they are all cis loop receptors, but there is more cis loop receptors that aren't GABA receptors, and another great family of ligand-gated ion channels which we're going to come across, which are cis loop ligand-gated ion channels, are the nicotinic acetylcholine receptors. Okay, so we will come on to these. Okay, after the benzodiazepines, we're going to study nicotine addiction. Okay, and that's going to be through action on the nicotinic acetylcholine receptors. Okay, and you'll be glad to know we don't need to go through a totally different receptor structure, because their receptor structure is almost identical to the GABA receptors, because both of them are within this great family of cis loop ligand-gated ion channels. Okay, so there are other receptors which are also in this family, such as the 5-HT3 receptors and also the glycine receptors that are used in the spinal cord. They're also within that family. A notable example of receptors that are not in this family are the glutamate ligand-gated ion channels. They have a very different structure to this. Okay, they're not pentamers for a start, they're tetramers. Okay, right, on to the GABA receptors then. So, we have discussed the structure of a GABA receptor subunit. It looks like this. Okay, now, unfortunately, there is not just one GABA receptor. It would be, there is not just one GABA receptor subunit. It would be lovely if there was just one GABA receptor subunit, and then to make a GABA receptor, all you had to do was make 5 of these GABA receptor subunits, stick them together, and you've then got your GABA receptor. It's not that simple in reality. Instead, there are actually 19 different types of GABA receptor subunits. Okay, 19 types of GABA receptor subunit. Okay, so I now want to tell you about the naming of these 19 different types of GABA receptor. Okay, so firstly, many of the GABA receptor subunits are grouped into families. Okay, so for instance, there is the alpha family of GABA receptors, and this contains 6 members, and they're very sensibly named. So there is the alpha 1 GABA receptor subunit, there is the alpha 2 GABA receptor subunit, there is the alpha 3 GABA receptor subunit, all the way up to the alpha 6 GABA receptor subunit. So beautiful, very easy to remember. Okay, next up there is the beta family of GABA receptor subunits. There is the beta 1 GABA receptor subunit, there is the beta 2 GABA receptor subunit, and there is the beta 3 GABA receptor subunit. So only 3 members in that family. It's the same for gamma. There are 3 members in the gamma family. You have gamma 1, gamma 2, and gamma 3. Okay, next up there is the Rho family. So Rho is the Greek letter that looks reasonably like a P. Okay, so there is 3 members of the Rho family: Rho 1, Rho 2, Rho 3. Okay, now we're getting into slightly more niche GABA receptor subunits now. Okay, the ones below the alphas, the betas, and the gammas, these are much more rare to see in GABA receptors, okay, but they are still GABA receptor subunits. Okay, these are the main ones, these are the notable ones. Okay, now, how many have we got up to now? We've done 6 here, 9 here, well, taking 3 more here, taking us up to 9, 3 more here, taking us up to 12, and then 3 more here, taking us up to 15. So we've got 4 more. The final 4 are not in families, or rather they're in families on their own. So there is then the delta GABA receptor subunit. Okay, there is the epsilon GABA receptor subunit. Then we go more dramatic Greek letters. We have the pi GABA receptor subunit, and then the theta GABA receptor subunit. So it's like Key Stage 3 maths again. Right, okay, so those are the 19 different types of GABA receptor subunit then. Okay, here they are. Okay, now I'm going to tell you the rules by which you create a GABA receptor then from these 19 different types of GABA receptor subunits, or at least I'm going to tell you the usual rules for creating a GABA receptor. Okay, because you will hopefully understand that there is an absolute nightmare potentially here. Because remember, GABA receptors are pentamers. They are made up from 5 GABA receptor subunits. I'll draw this just quickly here because it's an important picture and we've lost it by moving on to a separate piece of paper. Okay, so here is the full GABA receptor. Remember, the actual full receptor isn't just one of these GABA receptor subunits, it's a pentamer of 5 of them joined together. Okay, so what are the rules for joining GABA receptor subunits together? Okay, that's what we want to now ask. Okay, so generally, most GABA receptors are what are known as alpha, beta, gamma GABA receptors. Okay, and I'll tell you the rules now for constructing an alpha, beta, gamma GABA receptor. Okay, so what we are going to do is we're going to look down from above. So we're going to look at a GABA receptor from above. Okay, and what we will see is something that looks like this. Okay, like so. Here's the pore down the middle. Okay, and it's going to be made up of 5 separate subunits here. Okay, 1, 2, 3, 4, and 5. Okay, right. Now, basically, this is the rules for creating a GABA receptor pentamer like this out of these alpha, beta, and gamma subunits. So, firstly, what you do is you go to the alpha family of GABA receptor subunits and you pick one of those 6 members. Okay, so you pick one of the 6 members in this alpha family and you stick with that member. Okay, you make 2 copies of that member. So let's say I'm going to pick alpha 2 as my alpha GABA receptor subunit. Okay, I will then make 2 copies of alpha 2 GABA receptor subunits and I will put them here and here. Okay, so I'll colour this in orange. So here are the alpha subunits of the GABA receptor. One here, okay, and another here. Okay, right. Then what you're going to do is you're going to go to the beta family of GABA receptor subunits, okay, containing these 2 members, sorry, these 3 members, and you're going to pick one of those 3 GABA receptor beta subunits, okay, and you're going to make it twice, and you're going to put the 2 beta GABA receptor subunits here and here. Okay, so I'll colour these in green here. Okay, and these are the same beta subunit. Okay, so you can't put one beta 1 here and one beta 2 here. They have to be the same type of beta subunit. Okay, and then finally you'll go to the gamma family. You'll pick one of those gamma subunits and you'll put it here. Okay, right. And this now creates you the full alpha, beta, gamma GABA receptor. So most of the GABA receptors that are in the brain look like this, basically. They have 2 identical alpha subunits in them, they have 2 identical beta subunits in them, and they have a gamma subunit in them. Now, let me tell you about where the GABA binding sites are on this GABA receptor of this alpha beta gamma type. Okay, so basically the GABA binding sites are between the alpha and the beta subunits. So you only have 2 GABA binding sites per GABA receptor. You don't have 5 GABA binding sites. Okay, you only have 2. Okay, so these are the 2 GABA binding sites. Okay, so basically what will happen is you will have these GABA receptors on the postsynaptic membrane of your dendritic spine. Okay, so going back to this picture here, okay, you'll have these receptors on the dendritic spine here. GABA will then be released from the axon terminal of this GABAergic inhibitory interneuron here. Okay, it will then diffuse across the synaptic cleft, and 2 GABA molecules will bind to each of the GABA receptors on the postsynaptic membrane. Okay, when the GABA binds, it will cause the GABA receptor to open, so the pore will become much larger, and now chloride anions will actually be able to move through that pore. And chloride anions are going to move from the extracellular fluid into the intracellular fluid, and that's going to provide the inhibitory signal that's going to inhibit that postsynaptic neuron. Okay, now let me describe then the benzodiazepine binding site and where benzodiazepines are going to bind. Okay, and then I will solve the puzzle of how these benzodiazepines are actually going to raise dopamine level, because from what I've told you now, you might start to put this together in your head, especially if you know something about what benzodiazepines are going to do, and you might think, hang on, this is going to make dopamine go down, but you will see how it makes dopamine go up in a moment. Okay, so continuing on then, let's show where the benzodiazepine binding site is on the GABA receptor. So on an alpha beta gamma GABA receptor, the benzodiazepine binding site is located between the alpha and the gamma subunit here. So this is the benzodiazepine, which I'll abbreviate to BZD, binding site. Okay, right. Now, in order for the to actually be a benzodiazepine binding site, you have to have certain alpha subunits here and a certain gamma subunit here. Specifically, this gamma subunit has to be gamma 2. Now that usually is the case. Gamma 2 is one of the most popular gamma subunits for GABA receptors to have. Okay, the alpha subunit is not quite so picky. Okay, the alpha subunit needs to be one of alpha 1, alpha 2, alpha 3, or alpha 5. Okay, in order for there to be a benzodiazepine binding site there. So if you pick alpha 6, it doesn't work. Okay, you need one of these 4 alpha subunits here, and then of course there's the absolute requirement that you need to have a gamma 2 GABA receptor subunit in the gamma position there. Okay, right. So if you have a GABA receptor which does fulfill all of these requirements, i.e. that it's an alpha beta gamma GABA receptor and the alpha subunit is one of these and the gamma subunit is this one, okay, then what can happen is your benzodiazepine, so diazepam, chlordiazepoxide, lorazepam or clonazepam, can bind to that benzodiazepine binding site, and what it does is it causes a change in conformation in the GABA receptor, but that does not, repeat, that does not cause the GABA receptor to open. Instead, what it does is it causes a conformation that changes the conformation of the GABA binding sites. Okay, so we're now going to change the conformation of the GABA binding sites, and now the affinity of the GABA binding sites for GABA is going to be hugely increased. Okay, now what does that mean? Well, remember, in order for GABA to actually cause the GABA receptors to open, it needs to actually bind to these binding sites. Now, how hard does the GABA bind to the GABA binding site? Well, that is what we mean when we say the affinity. This means how much does GABA actually want to bind there? Okay, how strong is the bond between the GABA and the GABA binding site? Okay, because if it's very, very strong, then you'll get a huge amount of GABA binding to the GABA receptors. Okay, so what you have to remember is, if we draw our dendritic spine here, okay, we will have a huge number of GABA receptors. Okay, here's one, here's another one. We don't just have one GABA receptor on the postsynaptic membrane, we'll have a huge number of the things. Okay, and when you release lots of GABA onto the postsynaptic membrane, a fraction of the GABA-A receptors are going to end up with GABA bound to them. It's not necessarily going to be all of them, okay? Maybe, let's say, 30% of them end up with GABA bound to them, okay? Those are the ones that are actually going to open, okay? And of course you need 2 GABAs to bind you, okay? Those are the ones that are actually going to open and allow chloride in, okay? If we now expose this axon terminal to the benzodiazepine, okay, let's imagine that most of them get the benzodiazepine bound to them. Now what's going to happen is they're all going to have much stronger interactions with the GABA. Their GABA binding sites are going to have much stronger interactions with the GABA. So now much more GABA molecules are going to end up binding to the GABA-A receptors. So a much bigger fraction, let's say 60%, and I'm just making these numbers up off the top of my head to illustrate the principle, okay? A much bigger percentage of the GABA-A receptors are actually going to get 2 GABA molecules bound to them, and therefore a much bigger fraction of the GABA-A receptors are actually going to open, so you're getting much more chloride current now, so this is going to go up. Okay, so you're going to now inhibit neurons more, and this is where I said those of you who knew something about the way benzodiazepines would work might be thinking, wait a second, this is wrong, because if we're going to increase GABAergic neurotransmission, we're going to inhibit the dopaminergic neurons, aren't we? And that's going to reduce the amount of dopamine going to the nucleus accumbens and medial prefrontal cortex, which is not going to produce addiction, okay? But this is where it becomes more complicated, okay? Basically, let's draw a simple picture. Let's have the GABAergic neuron shown here, and I'm going to show it more simple now. I'm just going to show them as the cell bodies as circles with the nucleus in, like so, and then I'll have the axon shown as a line, like so, and then the axon terminal shown like this. So let's say that this is our GABAergic neuron. Okay, this is our GABAergic interneuron, so I'll colour it in blue here. Okay, and it's going to synapse onto our dopaminergic neuron here, which will send its axon to the nucleus accumbens or to the medial prefrontal cortex. So this is the dopaminergic neuron here, and of course everything we're talking about is happening in the ventral tegmental area. Okay, so here it is in red. Okay, and this is going off to either the nucleus accumbens or the medial prefrontal cortex, one of the two. Okay, now, the GABAergic neuron itself, okay, so this, remember, is the GABAergic neuron, is going to have other GABAergic neurons acting on it. Okay, so here is another GABAergic neuron. Okay, now, here is the thing. This GABAergic neuron here, which I'll also colour in blue, is going to be releasing GABA onto the GABAergic neuron that synapses onto the dopaminergic neuron. Okay, so you're going to be releasing GABA on here, and this is going to be trying to inhibit the inhibitor. Okay, so if this one became stronger, okay, and inhibited this one here, okay, let's give this neuron a name. Let's call this neuron 2, and we'll call this neuron 1. Okay, if neuron 1's GABA became stronger, if the effect of the GABA on neuron 2 became stronger, then that would inhibit neuron 2 from firing action potentials. Neuron 2 would stop firing action potentials, and therefore it would stop releasing GABA onto the dopaminergic neuron, and then the dopaminergic neuron's inhibition would go down, so it would now fire action potentials more. So you might now ask, but that's just silly. How do we know that this one's going to be potentiated by the benzodiazepine and this one isn't? Well, the reason is that this synapse here between neuron 1 and neuron 2 is going to have a different type of GABA-A receptor in it than this synapse here between neuron 2 and let's call it neuron 3. Okay? In between neuron 1 and neuron 2, you are going to have alpha 1 containing GABA-A receptors. Okay, so I'll write this here, alpha 1 containing GABA-A receptors, i.e. they're going to be of this alpha beta gamma type, and their alpha subunit is going to be the alpha 1 type. Okay, so in here we'll have 2 alpha 1 subunits, whereas in this synapse here you're going to have alpha 3 containing GABA-A receptors. Now, even though I said that both alpha 1 and alpha 3 you could have here and still have a benzodiazepine binding site. It turns out that most of the benzodiazepines that are actually used clinically end up having a much higher affinity for alpha-1 containing GABA receptors, i.e. where the alpha subunits here are alpha-1, over alpha-3 containing GABA receptors. Okay, so that means that you're going to get benzodiazepines working more effectively here than here, okay, which means that benzodiazepines are going to increase the power of this synapse much more than they're going to increase the power of this synapse, which means they're going to end up inhibiting neuron 2, okay, which will then fire less often, and that means that neuron 3 is going to have less inhibition and it's overall going to fire more often. Okay, right, so that then is how benzodiazepines are believed to cause an increase in dopaminergic firing in the ventral tegmental area, because the effect on these inhibitory synapses between GABAergic neurons onto other GABAergic neurons is stronger than the effect of the benzodiazepines on the synapses between the GABAergic interneurons and the dopaminergic neurons, because these synapses contain the alpha-3 containing GABA receptors, and these ones contain alpha-1 containing GABA receptors. Okay, and benzodiazepines prefer these sort of receptors than these ones. They much prefer binding to those ones than those ones. Okay, right, that then concludes how benzodiazepines are addictive. Okay, we'll call it there for this video. In the next video, what we're going to turn our attention to is nicotine, okay, and then we'll move on to the opiates.